All right, so diving in again today, and whenever we hear about a new drug hitting the market, it feels like this big moment, right?
Yeah.
But the truth is, that announcement, that approval, is just the tip of the iceberg.
Yeah, I mean, it's a journey, a really long and complicated one. Bringing a new medicine to patients, it's a whole process, tons of stages, and at every single step, there are crucial choices to make, paths to choose. And it's risky, right? Not every promising idea actually pans out.
It's true, and that's why we're here today for this deep dive. We're going to zoom in on a really critical turning point in drug development, that shift from early-stage human trials, phase 1 and 2, to the much bigger, really decisive phase 3 studies. That's where the rubber meets the road.
Exactly, and I think what makes this transition so fascinating is that it's all about taking those first glimpses of a drug's potential, the hints we get from those early trials, and using that information to build the foundation for these much larger, more complex studies. It's about taking a calculated leap forward.
And we've got so much to unpack, right? I mean, our sources cover everything from those initial discoveries in the lab to those first tests in animals to the nitty-gritty of how clinical trials are designed, to how drugs behave once they're in the human body, to the role of the big regulatory players like the FDA and ICH. It's a lot.
Yeah, there's a ton of information to sift through, but our goal today is really straightforward. What did we learn from phase 1 and 2? What are the big takeaways about a drug's safety and whether it even has a shot at working? And crucially, how do these early findings shape the way we design those pivotal phase 3 trials? Because in the end, it all comes down to one thing, making sure that if and when a new medicine gets approved, it's both safe and effective for the people who need it.
All right, so let's break it down. Phase I and phase 2. What are the absolute must-knows coming out of these early trials? Let's start with safety. In phase I, safety is the top priority, right?
Yeah, absolutely. And before we even think about giving a drug to a human, there's a huge amount of work that happens in the lab and then in animals. Preclinical toxicology studies, they're crucial, like a first line of defense. We're looking for any red flags, any signs that the drug might be harmful. And some of our sources, like preclinical toxicology evaluation, really emphasize this point. If a drug shows major adverse effects in animals, that's a serious warning. We have to proceed with extreme caution before even considering testing it in humans.
It's like our initial risk assessment, trying to anticipate potential problems before they arise. And these animal studies also help us figure out a safe starting dose for those first human trials, right? Kind of like finding a safe entry point.
Yeah, exactly. Researchers will do what are called dose-finding studies in animals, testing a whole range of doses to try and pinpoint that sweet spot, a dose that's likely to be tolerated by humans while still having the potential to be effective. It ties directly into the concept of the dose toxicity curve, which we see pop up in clinical trial design. Then, in phase I trials, the main goal is to figure out the safety profile of the drug. In humans, we're looking for that maximum tolerated dose, the MTD. This usually happens in a small group of healthy volunteers, although sometimes it's done in patients, depending on the drug and the disease.
And we saw in preclinical toxicology evaluation that if unexpected toxicities do show up, even in those early animal studies, it can throw a real wrench in the works. It
Yeah.
can mean needing more resources, delaying the whole development process, sometimes even bringing things to a complete halt.
It's true. Unexpected toxicity can be a major setback. It can lead to higher development costs, sometimes even the termination of a promising drug's development. It's a tough reality of the process.
Absolutely. So we've talked about the importance of safety assessments. But another crucial piece of the puzzle is understanding how a drug behaves once it's inside the body. That's where we get into pharmacokinetics, or PK, and pharmacodynamics, or PD, right? It's kind of like mapping out the drug's journey through the system.
Yeah, you got it. Pharmacokinetics is basically studying what the body does to the drug. It's about how the drug gets absorbed into the bloodstream, how it spreads throughout the body, how it's broken down, and eventually how it's eliminated. And we have this handy acronym, ADME, absorption, distribution, metabolism, and excretion. And multiple sources hammer home the point that understanding these ADME processes is absolutely fundamental. Think about it. A drug that's poorly absorbed, we say it has low bioavailability, might seem ineffective in later trials simply because it's not reaching its target in the body at high enough concentrations.
So we could actually miss out on a potentially valuable drug just because we don't fully understand how it's moving through the body.
Exactly. It's a real risk, and that's why those PK studies are so crucial. They help us figure out things like how long a drug stays active in the body and how often it needs to be given what we call the dosing regimen.
Right, the dosing regimen. And bioavailability, that's a key part of absorption, right? It's about
Yeah.
how much of the drug actually makes it into the bloodstream. And factors like how well the drug dissolves its solubility and how easily it can cross cell membranes, its permeability, those are super important here. We've seen this discussed quite a bit in our sources.
Totally. For drugs that are taken orally, bioavailability is especially crucial. You could have a drug that works amazingly in the lab, but if it's not absorbed well in the gut, it's not going to be very useful clinically. And that's where concepts like the rule of five come in. Medicinal chemists use these rules to try and design molecules that are more likely to be absorbed well.
And then there's the whole issue of how a new drug might interact with other medications a patient is already taking. We call these drug interactions, or DDIs. Understanding how a drug is metabolized, broken down by the body, is essential for predicting and avoiding these potential interactions.
Absolutely, that's a huge safety concern. We often focus on the liver, specifically these enzymes called cytochrome P450 enzymes, because they play a big role in drug metabolism. If we know how a new drug is metabolized, we can start to predict how it might interact with other drugs that are processed by the same enzymes. Now, it's worth mentioning that for biologics, these protein-based drugs, we don't usually worry as much about these P450 interactions because biologics tend to be broken down in different ways.
Right, so different types of drugs, different considerations. Okay, so we've covered what the body does to the drug, that's PK. Now, what about pharmacodynamics, PD? That's all about what the drug does to the body, right? It's about its mechanism of action, how it actually produces its effects.
Exactly. Pharmacodynamic is all about the drug's target in the body. What does it bind to? How does that interaction lead to a biological response? Hopefully a therapeutic one. Having a clear understanding of the pharmacodynamics is crucial for designing effective clinical trials and interpreting the results. For example, knowing that the drug trastuzumab, which is also known as Herceptin, targets a specific protein called HER2 in certain breast cancers, really helped researchers design trials to evaluate its effectiveness.
It all ties back to that target, to understanding the mechanism at play. All right, so phase I gives us that vital safety data and those initial PK insights. Then phase 2 starts to give us some early clues about whether the drug might actually work in patients and the people who actually have the condition we're targeting.
That's right. Phase 2 trials are designed to look at preliminary efficacy in a specific patient population, people with the disease the drug is meant to treat. And researchers often look at things like the objective response rate, which tells us what percentage of patients showed a positive response to the treatment. But we have to be cautious here. Even if we see some promising signals, we have to remember that these phase 2 trials are smaller. That means there's a higher risk of getting misleading results, both false positives and false negatives.
So even those encouraging early signs, they need to be interpreted carefully. And then we come to the big shift, the transition to phase 3. And all the knowledge we've gained from those earlier trials, the safety profile, the PK/PD characteristics, those early hints of efficacy, all of that becomes the foundation for designing phase 3 trials. It's like taking the blueprints from the first drafts and using them to construct the actual building.
I like that analogy. It's a perfect way to describe it. The data we collect in phase I and 2 informs almost every aspect of phase 3 trial design. For instance, the safety profile we've built up helps us decide who can participate in the phase 3 study, who might be excluded because of potential risks, and what specific safety measures we need to monitor closely throughout the trial.
And the PK/PD data, well, that's essential for figuring out the best dose or doses to test and the best way to administer the drug in those larger phase 3 trials. We're trying to find the treatment regimen that has the highest chance of working safely and effectively.
Exactly. We want to strike that balance between safety and efficacy based on what we've learned in the earlier phases. And then there's the preliminary efficacy data from phase 2. That data helps us define the primary and secondary endpoints for phase 3. These are the specific things we'll be measuring to determine definitively if the drug provides a real, clinically meaningful benefit to patients.
So we're taking those initial clues and turning them into concrete, measurable outcomes that we can track in those larger trials. But it feels like moving to phase 3 also introduces a whole new level of complexity, right? I mean, it's a much bigger undertaking, isn't it?
Oh, absolutely. Phase 3 trials are significantly larger, often involving thousands of patients across multiple locations, sometimes even different countries. And with that increase in scale comes the need for really clear, well-defined eligibility criteria. We have to be very specific about who can participate in the study to make sure we're studying the right group of people, people who are most likely to benefit from the drug and who are least likely to experience adverse effects.
So we're really trying to refine the study population to make sure we're focusing on the patients who are most likely to respond to the treatment. And the study design itself has to be incredibly rigorous. It's about minimizing bias, making sure that any effect we observe is truly due to the drug and not some other factor. That's where things like randomization and blinding come into play, right?
You got it. Randomization is all about making sure that the groups in our study, the group getting the new drug and the group getting the standard treatment or a placebo, are as similar as possible at the start of the trial. And blinding? Well, that's about preventing both the patients and the researchers from knowing who's getting which treatment. That helps reduce the potential for bias in how the results are reported and interpreted. It's all about maintaining objectivity.
So we're taking those extra steps to make sure the data is as reliable as possible. And it seems like by the time a drug reaches phase 3, the stakes are even higher. The expectations from regulatory agencies like the FDA are even more stringent. They're looking for rock-solid evidence of both safety and efficacy before they'll even consider approving a new drug.
It's true. Phase 3 trials are really the make or break moment. They're pivotal for getting regulatory approval. The data we collect in Phase 3 has to be robust, statistically significant, and it has to clearly demonstrate that the drug's benefits outweigh its risks for the intended patient population. And this whole process, the way these trials are designed, conducted, everything, is heavily influenced by the guidelines set by the FDA and ICH. There are specific regulations that dictate how these trials should be run to ensure the safety and well-being of the participants.
So it's not just about the science, it's also about ethical considerations, about patient protection. And speaking of guidelines, ICH-M3, for example, that gives us guidance on when we need to do those non-clinical safety studies in animals to support clinical trials. It's all part of building a strong foundation for those human studies.
All right, so we're building this comprehensive body of evidence, and eventually all of this data is submitted to the regulatory authorities for review. Can we look at a real-world example to see how this progression from phase 1 to phase 3 actually plays out? Yeah, the development of trastuzumab, or Herceptin, is a really good example. The early phase 2 studies showed some really promising activity in women with a particular type of metastatic breast cancer, HER2 positive breast cancer, and that early evidence, that signal of efficacy, was strong enough to justify moving into those larger, more definitive phase 3 trials. And in those phase 3 trials, the drug really proved itself. It showed a clear benefit for those patients, which led to its initial approval. But what's even more interesting is that the story didn't end there. Subsequent phase 3 trials explored trastuzumab in earlier stages of breast cancer and in combination with other therapies, which ultimately expanded its use and helped even more patients.
So those initial phase 2 trials, they were like a springboard. They provided the justification for the investment and the effort needed for those larger phase 3 trials, and ultimately that investment paid off, benefiting a whole lot of patients.
Exactly. And this idea of using early data to inform later stage development decisions is seen even earlier in the process. For example, the decision to move a compound like compound 27, also known as RO0505082, into clinical trials was based on a careful analysis of preclinical data looking at its potency in the lab and in animal models, its PK properties, and any initial signs of toxicity. That whole package of preclinical data helped researchers decide whether it was worth taking the risk and moving into those first-in-human phase I studies.
It's a step-by-step process, each stage building upon the last, using the information we gain to make informed decisions about how to move forward. And of course, this whole journey takes place within a very clearly defined regulatory landscape, right?
Oh, yeah, absolutely. The FDA, for example, has specific regulations in place like 21 CFR Part 312, which governs what are called investigational new drug applications, or INDs. And then there are international guidelines from ICH, like those related to good clinical practice, GCP, which set the standards for ethical conduct in clinical trials and ensure the quality and reliability of the data we collect. These regulations and guidelines, they aren't just arbitrary hurdles. They're there to protect patients and to make sure that only safe and effective drugs make it to market.
Right, they're there for a reason. And those OECD GLP guidelines, those are the good laboratory practice guidelines. And along with those ICH guidelines, they really help ensure that those preclinical toxicology studies are up to par, that they meet the standards needed to justify starting clinical trials in humans. It's all about building a solid foundation for those human studies. So we've covered a lot of ground today, really highlighted the crucial role of those early phase clinical trials, phase 1 and 2. It's clear that the knowledge we gain in those early stages about a drug safety profile, how it interacts with the body, whether it even shows initial signs of efficacy, all of that is absolutely essential for designing and running successful phase 3 trials.
Couldn't have said it better myself. Phase I and 2, they're like the reconnaissance missions. They give us the information we need to make smart choices about which drug candidates to pursue and how to study them in those later stage trials. Without a thorough understanding of the drug's behavior in those early phases, our chances of success in phase 3 would be a lot slimmer.
It's like trying to navigate uncharted territory without a map. You might stumble upon something interesting, but you're much more likely to get lost along the way. And this whole process, this multi-phase journey, all guided by a well-defined regulatory framework, it's ultimately about getting safe and effective therapies to the people who need them. That's the end goal.
And it really speaks to the scientific rigor and the commitment to patient well-being that drives drug development. It's a complex process, but it's a process with a purpose.
So as we wrap up, I want you to think about something. At the beginning of this journey, there's so much uncertainty, so much risk. How do those uncertainties get addressed as we move through those later phases, especially in those large-scale phase 3 trials? What does that tell us about the likelihood of a drug actually making it from the lab bench to the pharmacy shelf? It's something to ponder, something to really dig into. And if you want to learn more, I definitely encourage you to check out those FDA and ICH guidelines. They're publicly available, and they provide a fascinating glimpse into the rules and regulations that govern clinical trials. Thanks for joining us for this deep dive. It's been a pleasure exploring this with you.
Likewise. Always a good time to dig into the complexities of drug development.
Absolutely. Until next time.